This lecture is about the structure and function of the phospholipid bilayer. So the first thing we have to remember is what the bilayer is made of. So one of the primary components is our phospholipids. And we've talked about phospholipid function before, but to remind you, because it is very important for why the membrane can function as it does, a phospholipid has four main parts. One part that's very important is this bottom here, which are the hydrophobic tails. So you can see all the black's carbon and the grays are hydrogens. So these tails are made of carbons and hydrogens. Sometimes the tails are saturated, meaning that they end up straight. They have all single bonds and the max amount of hydrogens attached to them, to the carbon backbone. Sometimes they're unsaturated with a cis double bond here that causes a kink in the tail. And depending on the ratio of saturated to unsaturated tails, the membranes behave differently. So different phospholipids have different length tails and different saturated, unsaturated tails. Those two tails are attached to a glycerol molecule. So this glycerol has 3 carbons. The third carbon is attached to a phosphate group. Phosphate group has a negative charge. And then the phosphate group is attached to some variable group here. They've given the example choline in this picture, but that can be a number of different things depending on, and that really dictates what kind of phospholipid you have. So we can represent a phospholipid with this kind of structure, with a circle with 2 little lines coming off of it. The circle part is the hydrophilic head. So the phosphate group and the glycerol plus the modifying group, they all interact with water well. They are hydrophilic. Then the tails, carbons and hydrogens, we know those are nonpolar, which means these are hydrophobic. So A would be the hydrophilic head, B are the hydrophobic tails. So what does this hydrophilic head, hydrophobic tail allow the membrane to do? Well, the first thing to note is when you put a phospholipid into water and vigorously shake them, they will form spontaneously into a bilayer. So with the heads facing the water, because they are hydrophilic, they are hydrogen bonding dipole-dipole interactions with the water. And then the tails kind of hide on the inside because they're hydrophobic, and so you can see the carbons and hydrogens here. And basically, the water is excluded from the tails are excluded from the water, and so you get this core here that does not have very much water in it. And it's really this core of these hydrophobic tails that's going to give the cell one of its functions called selective permeability. So selective permeability, though, that is due to the phospholipids and the proteins in the membrane. So selectively permeable means that you can regulate the type and rate of traffic in and out of the cell. So not everything goes in and out at all, and it certainly doesn't go, most of it does not move without some kind of regulation. So here's a little chunk of our phospholipid bilayer, all of our phospholipids, and we have membrane proteins embedded in that phospholipid. We call this the fluid mosaic model. These are moving around very quickly. The phospholipids are primarily moving this way. Every once in a while they'll flip, and the proteins are studding the membrane. So depending on what type of proteins you have, that also dictates what kind of molecules get in and out of the cell and how fast. So different cells will have different phospholipid type components, and different cells will have different proteins embedded or on the edges of their membrane. So to regulate the amount of fluidity of the membrane, you can change the ratio of unsaturated fatty acid tails on your phospholipids to saturated fatty acid tails. So when your membrane needs to retain its fluidity, so like it's cold out, let's say, that's going to make the phospholipids move slower, and so it starts getting viscous, and you want to increase fluidity. So if you put the kinks in the tails, just like it keeps olive oil, the unsaturated fatty acid tails, it's going to keep the membrane more fluid as well. So this strategy is used by organisms in cold environments or during the winter. Plants and animals, fungi, they can all use this kind of method, as well as prokaryotes too. Saturated fats are going to pack together well, just like our butter, but in this case it's our membrane, so the membrane stays more viscous, so this would be possibly maybe a prokaryote that lives in hot springs or some other kind of hot environment, going to have a lot more saturated tails because it needs to maintain its viscosity. In animals, there's another option that has to do with a different type of lipid. It's a steroid called cholesterol. So cholesterol are these 4 rings fused together. And the ring structure is rather interesting because it's flat and it's stiff. When the fluidity is getting low, like it's getting cooler, the stiff structure keeps the phospholipids apart so it maintains more fluidity. When it's getting warmer and the phospholipids are moving a lot, the cholesterol is going to be rigid again, and it's going to retain some structure in the membrane to keep it less fluid. So basically it's like a moderator. Don't get too fluid, don't get too viscous, and the cholesterol is able to make that happen. Sometimes in the cell there's like these regions of lots of cholesterol. We call those lipid rafts, and they're basically units of membrane that stay together as kind of a floating mass through the rest of the membrane. And a lot of times they'll cluster different proteins in the membrane there that that work together, and so they will travel together on the moving phospholipid bilayer. Here's an example of a type of cell. We know this must be an animal cell because it has an extracellular matrix as well as cholesterol. So plants couldn't have the cholesterol in that membrane. So here, phospholipid bilayer, cholesterol, we have our proteins, 2 different types, peripheral on the edges, integral embedded in the membrane, sometimes transmembrane all the way through, sometimes just part way through. Some of those proteins have these sugars on them, the oligosaccharides, carbohydrates. They call those glycoproteins. You can think of it glyco, like glycogen, glucose. So they're the proteins with the sugars. Then we have glycolipids. Some of the phospholipids have sugars attached to them. On the outside is this extracellular matrix of proteins and carbohydrates, and on the inside of the cell, right under the cell membrane, is the cytoskeleton. These are actin microfilaments that are going to contract and relax and help change the shape of the cell by actually making the membrane protrude or go in or make it wiggly, so it can do all kinds of things. And the membrane's very fluid. Here would be an example of a transmembrane protein, an integral protein. You can see lots of hydrophobic alpha helices, the secondary structure of the protein. That's very characteristic of the part that would be interacting with the hydrophobic core. And we have part on the extracellular side and part on the intracellular side, the cytoplasmic side. And perhaps this would be some sort of protein where a movement could happen from the outside to the inside, or a signal could be transduced from the outside to the inside. There are 6 functions we're talking about with our membrane proteins. This unit mostly deals with the transport, so kinds of proteins that allow molecules in and out of the cell. There are also proteins that are embedded in the membrane that are enzymes, so they do chemical reactions. There are proteins in the membrane that connect to the cytoskeleton and or the extracellular matrix. This would be in animal cells. And they can help transduce signals into the cell. There is cell-to-cell recognition based particularly off of glycoproteins. So depending on the different types of carbohydrates attached to the protein, different cells have different signatures. So when 2 cells interact with each other, so this is one cell, this is the other, based off of the glycoprotein composition, in part, the cells can recognize each other and elicit certain responses. There is intercellular joining. You can see these 2 proteins are going to bond here and keep those cells tightly bound together. And then we finally have signal transduction, which is a receptor in the membrane that's going to receive a signal from the outside and transduce that signal to the inside of the cell, where a series of steps happen and there's some sort of cellular response. an interesting application of this receptor idea is with HIV. So HIV is a virus. It has an envelope on the outside and has these glycoproteins on the envelope. And those glycoproteins recognize on the white blood cells 2 different protein receptors. So they bind CD4 and they bind CCR5. And after binding those, the virus is able, the envelope fuses with the phospholipid bilayer, the virus enters the cell and then basically takes over the white blood cell, replicates many more copies of itself, and then goes on, buds out, and infects more cells. And so that would be when this happens too much, that's when you end up getting AIDS. They found some people had HIV infections, but they were not getting AIDS. They were living a long time, or just not getting any symptoms of that at all. So they did some studies, and one of the things they found was some people have a mutation in their genome that makes the CCR5 gene non-functional. So there's a mutation in the DNA code that when it's read, it doesn't code for the protein anymore. So you don't have any CCR5, the blue-green one. Apparently that doesn't have major consequences in the immune system because these are white blood cells. But what it does have major consequence of is that the HIV, when it binds, if it doesn't bind both protein receptors, then it can't enter the cell, and so it just leaves. So you can have this infection of the viruses, but they don't get into your white blood cells, and so that would mean that they don't replicate, and then they don't infect more of your cells. So there's some ideas that, well, what if we could make a drug that targets CCR5 in order to block it so that HIV isn't able to bind? You can't do that with CD4 because CD4 is essential for immune system function. That would kill the person anyway. But CCR5 is a possible target. So that's an overview of the structure and function of plasma membranes.